All right, we are in chapter 10, muscle tissue. So what we're going to be looking at in this chapter are the three basic types of muscles: skeletal muscle, cardiac muscle, and smooth muscle. Let's see if I can get my pen to work. There we go. Muscle is one of the four primary tissue types, and its job is to contract and allow movement of certain organs and items on the body. So, what are the characteristics of muscle tissue itself? Okay, it's excitable. It actually receives an electrical signal that allows it to respond to a stimulus. Now, the stimulus can be something from the environment, but the stimulus can also be a thought to induce movement of skeletal muscles. So skeletal muscle does respond to the nervous system. Cardiac and smooth muscle also respond to the nervous system, but it responds to the autonomic nervous system, not the voluntary nervous system. And so we're going to look at that in the second half of this unit, where we talk about the nervous system. We have the voluntary nervous system and the involuntary nervous system branches. Cardiac and smooth muscle is also very responsive to circulating hormones. And so we don't talk about that so much in these sections. The hormones that affect muscle typically are going to be like epinephrine, which is also known as adrenaline. Epinephrine also stimulates skeletal muscle, but it can change the activity of both cardiac and smooth muscle, especially during a fight or flight response. We talked about a characteristic of skeletal or muscle tissue in general, contractibility. That means that a piece of muscle, any type of muscle, smooth, cardiac, or skeletal, can become shortened, so it would scrunch up, or it can relax and elongate. Extensibility is that elongation, the extension of a muscle fiber after it's been shortened. And then muscle tissue has to be elastic, all right? You don't want muscle tissue to shorten up and then when it starts extending to rip. So elasticity is a big part of muscle tissue. So what does muscle do? Well, we mentioned it allows movement. It allows us to maintain posture and body position. The ability to stand is actually due to really strong muscles in your core. Even though I don't know how you think what sort of shape your core is in, but the core muscles, the longest muscles and the rectus muscles in the front and the anterior and posterior of your body are really strong. They remain contracted for hours if you're standing or even if you're sitting. So that takes a lot of strength and a lot of very high metabolism actually to maintain posture. Skeletal muscles also help to support soft body tissues. We'll get not into that too much, but muscles, some of the softer body tissues, tendons, ligaments, but even internally are suspended off of the interior muscles of the body. Skeletal muscle also helps regulate entering and exiting material. In other words, it regulates how food comes in and exits the body. Okay, so there are circular muscles that allow that to happen. Those are called sphincters. And skeletal muscle also helps us maintain body temperature. If you think about it, when you're cold, you shiver. Shivering are the really small contractions of muscles that increase your metabolism and increase your body temperature so that you're not freezing. So here's what the three types of muscles look like. Skeletal muscle, now I wish these pictures were a little bit clearer. If you go and you look at the histology slides that I posted individually in one of the files, you'll see that skeletal muscle has lots and lots of stripes inside of them, all right? Lots and lots of banding. They look like little zebra stripes, all right? And sometimes you'll see some little stripes like this going that way. Sometimes. Mostly you're going to see lots of zebra stripes. And in fact, they're much smaller than what I drew there in red. It's hard for me to draw them with this pen. So skeletal muscle contains lots and lots of little stripes, lots of little bands, and multiple nuclei. I don't know if you guys remember from one of the exams, I asked you if the cell has to go through cytokinesis after mitosis to survive. And this is an example where it doesn't. A single long muscle cell, and skeletal muscle cells can be really long, has multiple nuclei, and that's to allow protein synthesis along the entire length of the muscle fiber, all right? So a muscle cell can also be called a muscle fiber, and the length of a muscle cell is from the insertion to the origin. So if you think about your bicep, which starts way up in your shoulder and ends at your elbow, a single muscle cell extends from the insertion, from the origin in the shoulder down to the insertion in your elbow. And so that long, long cell needs more than one nucleus to produce messenger RNA so that protein can be made. Smooth muscle, which is over here in B, smooth muscle doesn't contain those striations, and it can have multiple nuclei, but the organization's a little different. We'll see that in other slides in a little bit. And then cardiac muscle actually does have the zebra band striping on it. You can't see it really well here. Has a single nucleus in each cell, but one of the characteristics of cardiac muscle is that it branches. And so this slide isn't the greatest showing that, but again, if you go and you look at that histology, series of histology slides, which I'll probably be pulling some of the identification pictures from, that or drawings for the next exam, you'll see the structure of cardiac muscle, and we'll get to that again in a little bit. So how is muscle organized? So if you look at a muscle, one of the things that you think about is if you think of somebody flexing their muscle, if you're thinking of the bicep, okay, the bicep has this nice round, what we call belly of the muscle, and that's the bump of the bicep. The bicep itself starts at one area at the origin with connective tissue. The connective tissue, which is going to be lots and lots of, which is the tendon here, is going to connect connective tissue to actual skeletal muscle cells. The muscle cells are going to be organized in what we call a fascicle. We'll get to all the details of each individual muscle cell in a few minutes, but let's start large here. So the whole muscle is surrounded by what we call epimyosin. The epimyosin is an extension of that fibrous connective tissue that seals up the entire muscle, okay? The epimyosin encircles and encapsulate individual muscle fascicles. So the fascicles are smaller groups of muscle fibers, okay? And we'll get to what that is in a minute. Each fascicle here is surrounded by perimyosin. Perimyosin connects to the epimyosin, but you can think about the perimyosin as kind of an extension of that epimyosin, separating out clusters of muscle fascicles, okay? Now, when you take this whole fascicle here, this circle, the fascicle then breaks down into individual muscle fibers. Each one of these circles inside of the fascicle is a fiber, okay? And that fiber is the individual cell. That cell is surrounded by the endomyosin, which is connected to the perimyosin, which is connected to the epimyosin. So you have an extension here of connective tissue starting from the outside down to the level of the muscle cell itself. This is a little satellite cell. It's a cell that kind of goes and cleans up the environment for the individual muscle fiber. Now, what are these little tiny dots in here? These are called myofibrils. This is where the actin and the myosin is, okay? And we'll get to that later, what they're doing. You can kind of start to see right here, the zebra banding inside of a myofibril. So we start with muscle, then fascicle, then fiber, then fibril, okay? So I think about skeletal muscle as being the largest level, the muscle fascicle being slightly smaller, the muscle fiber being the actual cell, which is made up of myofibrils, okay? So this is starting top to bottom, or largest to smallest. And all of these little myofibrils merge at the tendon. The tendon, the aponeurosis of a muscle is the aponeurosis is the connection of the muscle to the flattened portion of the tendon. So this right here is the aponeurosis. Let me erase that. That doesn't look so good. Let me push the eraser button there. That's the aponeurosis. And actually, to be honest, it kind of extends even farther down. Oh, stop it. It extends farther down. My pen is just misbehaving today. All right, the white part. So I included this second picture here so that you can look at the organization of muscle, starting at the tendon or the aponeuroses, the epimyosin, underneath the epimyosin, and between, within, I guess you would say, that space where the perimyosin is, and between the skeletal muscle fibers is where you're going to find nerves and vasculature. Remember that muscle is extremely, extremely, extremely metabolically active. And what that means is you have to have lots of oxygenated blood there to supply the muscles with oxygen so that they can make and use ATP. So when we started over here, here's our muscle, the organ, that's level 1. Level 2 is the fascicle, remember. Level 3 is the muscle fiber. And level 4 is going, well, the muscle fiber, muscle cell. And inside of that, you're going to have all of the little myofibrils, okay? Remember that? Number 4, let's go back to the last slide. Myofibril, right there. Myofibrils here, all right? Smallest level here, which contains actin and myosin. So when we do a cross-section through the muscle, you can see individual fascicles, remember, level 2. You can see the epimyosin, the perimyosin, right? And the endomyosin, sort of, surrounding each little fascicle, little group. And then you can see where you see clusters of vasculature and nerve innervation. So let's look at the muscle fiber. This is a picture from your book. I like this picture because it's really well defined. It has a lot of elements that you're going to need to be able to identify on the next exam and answer questions about on the quiz. Usually we have our quizzes on Sunday. What I'm going to be doing is doing a quiz over chapter 10 and 11 at the same time. So that's going to be on next Sunday on the 18th. So, and then for at least fall 2020. So what we're going to be looking at here is the muscle fiber. The muscle fiber is broken down into the filaments, the thick filaments, which are made of myosin, and the thin filaments, which are made of actin. Now, there are different versions of actin. Don't worry about that right now. There's F-actin and things like that. What's important about this particular picture is that you can see that the myofibril, when you enlarge it, contains, like I said, those little zebra bands. The Z disc and the I band are a region where you have a protein called titin, which is going to be connecting to the myosin. The myosin is the thick filament, the thick dark purple lines. And then the actin filaments are these thinner green filaments here attached to the Z disc, okay? And then titin is going to be associated with myosin. It's kind of actually over here. There's another protein here. So what you're looking at is what we call the sarcomere. The sarcomere are the alternating units between actin and myosin, green and purple, and how actin and myosin are anchored and how they overlap with each other results in what we call the different bands, the I band, the A band, and the H zone. Sometimes some people call that the H band, and then you have the M band over here. I like the M band because I think of it as the myosin band, and that's what's anchoring myosin in place. The Z disc is inside of the I band, which is anchoring actin in place. Over the top of the whole muscle filament, okay, or the whole fiber here, is this other organ called the sarcoplasmic reticulum. Now, this normally would be overlying this entire region here. The reason why it's been pulled away is so that you can see the different bands and discs, all right? The sarcoplasmic reticulum carries and stores ionic calcium. This is one of the places in the body where calcium is essential. Your muscles will not contract without calcium and magnesium, all right? Those two ions are essential. So if you want to have nice, strong muscle contractions, and if you like to work out, if you like to lift heavy things, or you like to do lots of cardio training, you have to consume adequate amounts of calcium and magnesium so that your sarcoplasmic reticulum has plenty of calcium stored inside of it, and that there's enough magnesium circulating around in your bloodstream to supply magnesium for the contraction. You'll learn more about that in physiology. All right, so let's look at a little more detail about the muscle fibers themselves, not so much the organization of the sarcomere. Myoblasts are the early muscle cells that congregate and develop into a multinucleated muscle. So they literally migrate together and start fusing initially, okay? So the myoblasts produce individual or contribute their individual nuclei to one muscle cell. Look at that, isn't that cool? These nuclei can divide they can undergo mitosis as we grow or as we work out. Don't worry about that right now. The sarcolemma is the plasma membrane, okay, of the cell. The sarcoplasm is the cytoplasm of a muscle cell. All right, so they're showing you here in this picture the sarcolemma and the sarcoplasm. And the sarcoplasmic reticulum, which I mentioned before, stores a tremendous amount of ionic calcium. Now, the sarcoplasmic reticulum is connected to another organ called the T-tubule. The T-tubule is going to be nestled between these repeating units of sarcoplasmic reticulum. And in this particular picture, you can see how the sarcoplasmic reticulum would extend from one sarcomere to the next sarcomere to the next sarcomere, all the way down the length of the skeletal muscle. Now, the T-tubules themselves are an extension of the sarcolemma, all right? Basically, it's a place where an electrical signal travels. You'll hear a lot more about that in physiology. The triad consists of the T-tubule and the sarcoplasmic reticulum on both sides. So here's triad right here, okay? It's just this plus this plus this, okay? The myofibril itself is the combination of your thick and thin filaments. So it's thick plus thin equals that, all right? And then, like we looked at in the last figure, the sarcomeres are the functional repeating units of actin and myosin organized in such a way that actin and myosin overlap. All right. We spend a whole lot of time on sarcomere organization. We did initially cover that a couple slides ago, but let's look at it again in this organization because it shows us, this picture is going to show us a cross section of what each of these bands look like in a sarcomere. So our thick and thin filaments, myosin and actin, the Z lines define the end of the sarcomere, all right? So that's our Z line right here. The A band contains thick filaments and might include some thin filaments. You know what? I should have just circled this. Here's the Z line, but here's our Z line. Here's our A band right here. Okay. Within the A band is the H band right here. It's kind of the center of all the myosin molecules, and you're never going to see actin there. The I band is thin filaments only, and then the M line is what holds myosin in place, all right? And they don't show that here, or here for that matter, which is kind of weird. It's the center of the H band, all right? So, oh, it's right here. Here's the M line, center of the H band, okay? Oh, here's the M line. I just missed it. Sorry. Here's a blow up of the sarcomere organization, I band, A band. So this is a structure that you're going to need to know for the exam. What happens? Now, I'm not going to go into great detail of what a contraction looks like molecularly. All right, you're going to learn that in physiology, or you've already learned that in physiology. But the idea is that as calcium is released from the sarcoplasmic reticulum, these little bumps on the myosin, they're called globular heads. These bumps grab onto the actin and pull actin inwards to shorten the distance between Z line number 1 and Z line number 2. So a contraction is a shortening of the sarcomere. And that happens when electrical activity induces the release of calcium from the sarcoplasmic reticulum. Now, when the sarcoplasmic reticulum releases calcium, I'm not going to go into details, but basically what happens is that actin and myosin are able to get together. Here are these globular heads, tighten filaments holding myosin in place, and that shortening is the result of what we call a power stroke, where the myosin head bends over, which is what pulls actin over the myosin heads. I am not going to ask you how that happens in the exam, all right? But I do want you to know the organization of a sarcomere and kind of what the functional part of the myosin, which is the myosin head, which pulls actin across it as a contraction is occurring. All right, we're going to move on to just a little review of how that electrical activity arrives at a skeletal muscle cell. Now, on this picture, on A, you can see the beautiful, beautiful sarcomere structure, or the zebra banding, in these little pieces of skeletal muscle. Over here, this is an electron micrograph, a scanning electron micrograph, so you can't see the bands because those are internal, okay? You'd need a TEM to see that. This would be an axon of a neuron extending down and then to touch in this case, this particular muscle cell. The place where the axon innervates or touches the muscle cell is called a neuromuscular junction. It literally is this complex network of feet touching the skeletal muscle fiber. Each skeletal muscle fiber should have at least one axon touching it to tell that muscle fiber it's time to contract. Depending on the nerve itself, the nerve may have many, many, many axons touching many, many muscle fibers, or just a few axons touching muscle fibers. You'll learn more about that in physiology and how what we call the innervation ratio, the number of axons to muscle fibers, allows you to make really big, strong muscular movements or really fine, sometimes even slow muscular movements for fine coordination versus rough coordination. So it's different running, right? When you're using your gluteal muscles, let's say, really strong muscles, they just need to contract and fire so that you can move fast. That's a different type of muscle contraction and muscle utilization than writing with your fingers, right? Your fingers have to be well coordinated. That's fine muscle movement. And so you have a different innervation ratio for that. Now, I mentioned that the control for a muscular contraction starts with, or at least the initiation of a muscular contraction starts when the sarcoplasmic sarcoplasmic reticulum releases calcium. The calcium is going to interact with the muscle fibers themselves to allow contraction to occur. How does that signal get to the sarcoplasmic reticulum, though, right? So what you have is a series of motor neurons in the spinal cord, in the ventral horn of the spinal cord. We'll talk about that later on in this unit. Those motor neurons, they're called motor because they interact with skeletal muscle, which allows you to move. One motor neuron contacts a certain number of muscle fibers. So in this case, the purple motor neuron is innervating 1 and 2 fibers. We call that a motor unit. So the neuron plus the fibers that it contacts is a motor unit. So the blue neuron is innervating these blue regions, and the red neuron is innervating these red regions. Okay, so the red has 1, 2, 3, 4. The blue has, looks like 3, and the purple has, it looks like 2. Okay, so motor unit number 3, which has only 2 muscle fibers associated with it, has less strength than motor unit number 2, which has 1, 2, 3 muscle fibers, which is weaker than, or slightly weaker than, motor unit number 1, which has 1, 2, 3, 4 muscle fibers. Okay, number of fibers. So what that means is that motor unit 1 can produce a stronger contraction than motor unit 3. It has 4 fibers to do work, whereas motor unit 3 only has 2. So this is one way that the nervous system is able to control how strong a contraction is produced. If you just need to pick your arm up, let's say, your hand up to scratch your nose, you don't want a really strong contraction. You'll punch yourself in the nose. So a weak contraction, you might use just the red motor neuron to activate the muscle contraction, your biceps brachii, right? If you need something a little stronger, let's say there's a mosquito on your forehead, you want to slap your forehead. I don't know if you could hear that slap on my forehead. You might use motor unit number 2. Motor unit number 1 would be for stronger contraction. All right. Now let's say you're trying to pick up a real heavy weight. You might use all 3 motor units and maximally contract that muscle. That's basic physiology. There's more detail to this. It's really an elegant system that produces very, very smooth contractions based on the number of motor units that are activated given the job that needs to happen. And your brain plans all this out exquisitely, but I'll let you guys learn about that in physiology. The end of the neuron right here, these little dots, are what we call the synaptic end bulb. They're filled with acetylcholine. These little acetylcholine vesicles, remember we looked at this little flower spray at the end? In fact, that's what they call it, a flower spray. Actually digs down into the muscle, and the muscle cell itself has these little zigzags called junctional folds. On the little junctional folds are acetylcholine receptor sites, and this little lunate shape is called acetylcholinesterase molecules. Acetylcholinesterase breaks down acetylcholine, which are in these little red circles inside of vesicles. So when a contraction is triggered by the motor neuron, the electrical activity arrives at the motor end plate, allowing the release of acetylcholine into the synaptic cleft. This is called the cleft, okay? It's just the distance between the motor end plate and the muscle. As the acetylcholine drifts across, some of the acetylcholine lands right here onto the receptors, but some of it interacts with the acetylcholinesterase and it wipes out the acetylcholine. It breaks it down. This is a good thing, all right? If acetylcholine remains in the cleft, or if you have too much acetylcholine in the cleft, it does start a muscle contraction. It starts the myofibrils and the actin and myosin moving, all right? It induces calcium release. You'll hear about all that stuff in physiology. But the acetylcholinesterase helps control how much of that happens and how long it happens, all right? If you do not have acetylcholinesterase, that acetylcholine will remain bouncing around in here and continue activating the muscle, and you'll end up with extreme muscle cramps. I believe that it is either cobra or rattlesnake venom. I think it's rattlesnake venom that wipes out acetylcholinesterase. And what that does is that it causes contraction of muscle, but the muscle can't relax because it continually becomes activated, which means that if acetylcholinesterase is inactive, your muscles, as they don't relax, if that happens to like the muscle in your respiratory pathway, you'll breathe in, but you won't breathe out. So acetylcholine is a big deal here. This is just a slide showing you the release of acetylcholine, allowing the electrical activity in acetylcholine to travel down the T-tubule. That causes this is the action potential, okay? The action potential spreads, releasing calcium from the sarcoplasmic reticulum, allowing the contraction to occur, shortening the sarcomere. And this is relaxation. So this is contraction, and this is relaxation. And so here's a few pictures from your book. I pulled these pictures from a different book because they just looked clearer to me. But in your book, you can go to figure 10.6 and look over the basic signaling mechanism for acetylcholine and the acetylcholine receptor. And just so you know, acetylcholine does not go inside of the muscle cell. It allows sodium to go inside. But again, that's physiology. And then here's the sarcoplasmic reticulum and the triad. So your electrical potential comes in here, allowing calcium to be released from the sarcoplasmic reticulum. And then these are just more pictures showing you contraction eventually at the muscle level. And then relaxation is when calcium is pumped back into the sarcoplasmic reticulum and actin and myosin no longer interact with each other. And usually the opposite muscle, which we'll talk about in the next chapter, will contract, allowing the lengthening of the muscle that just contracted. All right, so that would be the center, the belly of the muscle. When the muscle relaxes, it's not necessarily relaxing on its own. It's antagonist, it's opposite. So if this was the biceps brachii, the triceps brachii would then, or the biceps would contract while this muscle relaxes and lengthens. So this is called the sliding filament model. This is contraction here. This is relaxation here. Okay, so that's C and that's R. And again, more examples of skeletal muscle contraction at the molecular level. It's physiology for you to review. I'm not going to ask you guys great detail on power stroke, the order of events on the exam. That's physiology. I am going to mention very briefly muscle metabolism. Muscles are very, very hungry. They use a tremendous amount of glucose and glycogen, muscle glycogen. You know what's funny? This is, one of the things that that cracks me up is if I go and I work out, I either lift or especially if I go running, I burn up a whole lot of muscle glycogen. And what that means is I'll lose I can lose up to like 2 to 2.5 pounds if I run like 6, 7, 8 miles. All right. That's a lot. I'm not losing any weight at all. Water follows glycogen. If you have lots of glycogen stored in your muscle, you're going to store a lot of water as well. That's all water weight. So when you burn up your glycogen, you haven't lost any fat. You certainly are not going to lose any fat in one run, right? That's just purging, that's just using up muscle glycogen and losing the water associated with it. So one of the things that happens when we're resting is ATP will come into contact with a compound called creatine. Some of you lifters might recognize that, to produce creatine phosphate. Creatine phosphate is a way to store phosphate molecules for muscle contraction. Don't worry about what's going to happen with that. That's physiology again. That releases ADP, and ADP can be recycled to ATP again via glycolysis, okay? So in glycolysis, you make 2 ATPs, you make pyruvate, and pyruvate is then going to produce lactic acid in the blood. Blood glucose, when you're doing a lot of metabolic activity, blood glucose does go down. You eat through that pretty quickly. Which is a good reason to eat, like an hour or two before you do a high metabolic cardio exercise for 40 minutes or so. Eat oatmeal or something because that keeps your blood glucose somewhat stable, all right? What's happening is that blood glucose is getting converted to lots and lots of ATP, heat, and CO2 through cellular respiration. Your body can also take pyruvic acid that's built up in your bloodstream or fatty acids. So believe it or not, one of the first things your body uses when it runs out of blood glucose is fat. That's going to be your circulating triglycerides in your bloodstream. Now, in terms of muscle contraction, there are a couple different types. There's isotonic and isometric. Isotonic is not the same thing as isotonic fluid. Oh, breathing. Sorry, guys. Sleepy this morning. During isotonic contractions, what's happening is the muscle is shortening. So isotonic contraction allows basically a concentric phase, which is a shortening of the muscle, and an eccentric phase, which is a lengthening of the muscle. All right, those are isotonic. Isometric contractions happen when the muscle contracts, but the length of the muscle doesn't change. There's no movement. So you can do isometric exercises, just holding a weight. That does allow shortening of the muscle. But even though the muscle shortens, the lever angle of your arm doesn't change. And so what's the ideal length of a sarcomere? Well, it depends on what you want to do, all right? The length is going to be small when a contraction has occurred, and the length of the sarcomere changes as a percentage of the sarcomere length when no cross bridges are formed or when you have relaxation. Okay, so this is contract. That's completely relaxed. And sometimes, usually during an isometric contraction, the length is in between. So now there are a few different types of muscle fibers. And some of you, again, if you work out, you'll know about this. There's fast twitch and slow twitch. In other words, white meat and dark meat. White meat or white muscle is our fast twitch, well, it doesn't, the white muscle fatigues quickly. It's usually used for high contraction rates, sprinting. It's different from slow twitch muscle. Now, what's confusing about this slide is there should be a space right here. So white muscle or white meat on your turkey allows for short contractions, okay? Basically, it's, you find a lot of white muscle in individuals who do a lot of lifting. Slow twitch muscle, on the other hand, is the red meat that you usually find in like chicken legs, turkey legs. It's red because it has another molecule called myoglobin in it. And myoglobin stores oxygen. It holds on to it. Myoglobin is similar to hemoglobin, right? But it remains in the muscle holding on to that oxygen because when you're, let's say, running away from the tiger who can chase you for a long time, right? Or when you're doing highly metabolic exercise, that's when you need lots of extra oxygen around, and the supply of oxygen coming from the hemoglobin in your bloodstream, that can't supply your muscles fast enough with the amount of oxygen it needs to make ATP to do what it needs to do over the long haul, all right? So myoglobin helps maintain oxygen. It's a buffer, basically. It's an oxygen buffer. So white meat, short bursts of high contraction, really fast contraction. Red meat, this is basically your endurance muscle. So this is how they look. The slow fibers are smaller in diameter, okay, darker in color because they are full of myoglobin, and fatigue resistant. You have to train them to be resistant to fatigue. But the slow twitch fibers you're going to find also in postural muscles, the deep muscles of the core, okay, the rectus muscles. Those muscles have lots of myoglobin in them because they need to have a constant feed of oxygen. The fast twitch muscles, on the other hand, the things that you find in the biceps, all right, larger, they look much more pale because they don't have myoglobin, and they are easily fatigued. Now, ideally, what do you want? You want a mix of both, all right? So one of the things that a lot of people notice is that in marathon runners, how do these guys look? Do they look buff? Well, they look lean. Okay, I'll give you that. You can see, look at that, sartorius. Dang, man. Sorry, I'm just always impressed by, people who are walking anatomy models. Long distance runners have lots and lots of that slow twitch muscle, and relatively few fast twitch fibers in their legs, all right? And in fact, that's something that is genetically predisposed to some people, where they say that the athlete doesn't pick the sport picks the athlete. Basically, you end up being good at something that you are predisposed to. So a lot of you don't know me. I'm a runner, okay? I've always been good at it, decent at it. I'm only 4'10.5", so I have a hard time beating people taller than me. But given my size, I'm actually pretty fast. My size and age, actually, I do a pretty good job. And when I did my genetic background, I found that sure enough, I was predisposed to having an abundance of slow twitch muscle fibers, which makes sense. So I have really strong legs and I can run a long way. Relatively few fast twitch muscles. I am not a great sprinter, okay? I can run a long distance for a long time, okay? I can beat a lot of people over the long haul. But 100-yard dash, no go, okay? Everyone's going to beat me, probably partly because I'm short too, but what are you going to do? So my slow twitch muscles far outweighs my fast twitch muscles. So what's going to make me faster? Okay, I can run and run and run and run, do lots of distance training, but that slow twitch is not going to get any better. The only thing I can do is train my fast twitch fibers, which means lifting. And so changing the density or the size of my fast twitch fibers has made me faster because I'm able to make these thicker and more resilient so that as my slow twitch muscle starts kind of petering out a little bit, some of the fast twitch can take over. It's a pretty cool system if you ask me. So bodybuilders contain lots of fast twitch fibers, right? Not as much slow twitch. Yet another walking anatomy example here. Wow. Hypertrophy is the process by which we make our muscles larger. When our muscles grow, you don't get more muscle. what you're doing is you are actually growing the size of each individual fascicle. You're adding actin and myosin. You can add actin and myosin to slow twitch, just like you can to fast twitch. It's just fast twitch tends to grow faster. Don't worry about why. That's physiology. So what is muscle tone then? Muscle tone is a partial contraction of those muscles. It's that intermediate length, right? It doesn't produce movement. Some of the fibers are relaxed, some are contracted, and it's important to keep good muscle tone to maintain your body position, to maintain your posture. Bad muscle tone, you're going to slouch. And so muscle tone also has to do with how much and how healthy muscle you have. And normal muscle, it'll be a certain size and be held at a certain tension. As we age, it's possible for muscle to become what we call atrophied. That happens when you lose actin and myosin filaments and the muscle gets real skinny. So muscle atrophy happens due to aging. It can be happen due to disease. It can happen through misuse. So if somebody breaks their arm and has to wear a cast and they're not able to contract skeletal muscle, it'll atrophy to some degree because it just hasn't been used. Now we're just going to briefly cover the last two types of muscle, cardiac muscle tissue. It's only found in the heart. Cardiac muscle tissue is found in the walls of the heart. It's striated. It is involuntary muscle. Skeletal muscle is the only type of muscle that's voluntary, all right? It is uninucleate. In other words, there's only one nucleus per cell. It has branches. So if we can find one here, here's one that branches. You see a leg there and a leg there. So this would be the muscle fiber here. I'm sorry that this is in red. I'll show you another picture in a minute. And then you also see these lines here. These are not a part of the sarcomere. These are called intercalated discs. And intercalated discs has gap junctions. Remember those? Which allows ions and an action potential or an electrical activity to move from one cell through to the next. So this is an artist rendition of a cardiac muscle fiber right here. So you can see the branches. And these are the intercalated discs. At the intercalated discs, you have quite a few desmosomes. And these are the gap junctions, allowing stuff to diffuse from one cell to the other. Okay? I'm sorry, those are desmosomes. My bad, I didn't read this figure. Oh my gosh. These are the gap junctions. So you have information flowing this way. I'm sorry. Why did they flip this? That is insane. Okay. I'm sorry. I was looking at information flowing this way, and that's why I did that. Okay. Smooth muscle organized a little differently. Okay. Smooth muscle does also have active innervation, as does cardiac muscle, but they're not showing it here, right? There is innervation. And smooth muscle is organized more like a net. So it's found in the digestive, urinary, reproductive organs. It's non-striated, uninucleate again, only single nucleus. It's involuntary. Cells taper at the ends. No striations are present, and that's why it looks smooth. It does have lots and lots of thin filaments, but they're arranged differently. So these dense bodies anchor myosin. These would be like in the place of the Z lines, all right? Actin and myosin and sarcomeres are organized like a net, all right? So when the muscle contracts, the net is basically pulling in on the muscle cell. And so how does the muscle know to do this? The nerves on smooth muscle are organized differently than they are on skeletal muscle. We're going to go into more detail when we hit the nervous system. But basically, the nerve runs through and makes numerous little points of contact, numerous little these are numerous neuromuscular junctions called varicosities. So this is the same thing as we saw at skeletal muscle. It just doesn't end. It just goes along the length of a band of smooth muscle. And so the varicosities contain the acetylcholine. Some of them contain acetylcholine. Some of them contain epinephrine, and some contain norepinephrine, depending on the branch of the nervous system you're talking about. And that contacts or instructs the muscle fibers to shorten. All right, that's it for chapter 10.